Hi everyone, in this video I'm going to review DNA replication in addition to the enzymes that you learned in class. This is a great practice question that is typically put on the exam with the orientation being flipped a little bit or slightly different, but this is a good practice question and I highly encourage you practice this, flip it around, figure out if you can identify the functions of helicase, topoisomerase, SSBPs, where the leading and the lagging strand would be found, where the Okazaki fragments would be found, and kind of just orienting yourself throughout this kind of whole diagram. So the first thing I'm going to start with is, in this example, the 5' and the 3', 5' on top, 3' on bottom. This can be flipped, so definitely practice it with 3' on top and 5' on bottom, just so you don't get used to practicing only one way. So the first thing that I'm going to go in label is topoisomerase. Topoisomerase is also known as gyrase in prokaryotes, but is more commonly known as topoisomerase in eukaryotes. It relaxes supercoiling, so it helps to prevent twisting and coiling by relieving tension. It quickly snips the backbone and then it immediately fixes it. It's kind of like magic. So in order for this to happen, it needs to be situated upstream because it's really hard for DNA replication to happen if everything's all mangled and tangled up. You need to make sure that everything's pulled tight and smooth to make sure that DNA process can kind of flow nicely. So again, this is where I would label topoisomerase. And again, it prevents coiling. And I will also have an answer key at the very end of this video so you can both check yourself and double check that you identified the specific roles of each enzyme correctly. The next thing I'm going to move on to is helicase. And helicase disrupts hydrogen bonds and helps with the unzipping process. So it has to be also situated upstream here at the top, not as far as topoisomerase, because again, topoisomerase needs to untwist and prevent the supercoiling, and then that allows helicase to disrupt the hydrogen bonds and open up the rest of the strand. So it disrupts hydrogen bonds. So a good way I like to remember these two is that topoisomerase is situated at the top or upstream, kind of this whole diagram. So again, situated at the top or the beginning. And then for helicase, I think of it as hello helicase, H and H. So, for example, if you had a greeter that was opening the door saying hello, they would make sure to, again, open up the whole thing. Okay, the next thing I want to note is that the 5' and the 3' right here, these are your parent strands. You don't have to label these. Some people like to, just to help orient themselves and remind themselves that these were the original parent or original strands that were given that you're working with. So I'll label them just for clarity's sake, but again, you don't have to label them if you don't want. So moving on, because we're working with the 5', 3', so I'm going to label the other two ends. So I know that the 3' would be on top because the 5' is right here. And then its mate for 3' again is 5'. So you'll have them be opposites. The next thing I want to show you is the origin of replication. That would be situated right here. So basically what I want you to imagine is that this is 1/2 of the replication bubble that we practiced in the first in-class assignment. So remember you guys worked with like a bubble like this. not a very good bubble, but you guys worked with a bubble like this, and then we split it in half. You're basically looking at this half of the bubble. This half right here is off the paper and you don't see it. So again, you're just looking at this half of the bubble when you're doing this particular assignment to help you kind of visualize like what you're even looking at. So again, this is where your origin of replication would be. Kind of on that first assignment, how we had that dotted line, this is exactly where that dotted line would be. Oh, that's... So now that you have your origin of replication, let's identify the replication fork movement, which would be going in this direction. And you know that because if you are opening, if helicase is opening everything going in this direction, and this is technically eventually going to get wider as new nucleotides are added, it will be going towards that left side. But once more, show you again, origin of replication, where when we drew it the other time, we showed that the arrows moved in opposite directions. Actually, it's really hard to see, but that the origin of replication, we're working on this side, so that's why it's moving towards the left. So I'll label that really quick as your replication fork movement. And then that will help us set up our 2 new daughter strands. Daughter strands are just the new strands that are being created. These are also your leading and your lagging strands. So leading strand, remember when we're labeling these, they will be opposite. So if 3' is right here, I'm going to draw a 5' right here and a 3' right there. And then for my bottom strand, we have a 3' right there, so this will be 5' and this will be 3'. A good way of checking yourself, a 3 and a 5 will always be next to each other. You will never have a parent and a daughter strand both be 5 or both be 3 right next to each other. Remember, they are opposites. So now that we have the replication fork movement, like the direction of it, you want to identify your 3' going in the direction of the replication fork. So that would be right here, this green 3', because it's going towards the left, which is the way that this piece of DNA is basically opening, or this replication is opening. So this right here would be your 3', which means 3 leads. So label this as leading, which means that your bottom strand would be lagging, going the opposite direction. And you can draw these however you'd like. Just make sure it's not a continuous piece. And then also you want to identify your Okazaki fragments. And remember that your Okazaki fragments are just those kind of like little pieces of DNA, like the little segments. That's why the lagging strand is also called the discontinuous strand. So this is your lagging strand. And then you can draw either little squiggles at the end or do however you would like. But in some way, make sure to label your Okazaki fragments somewhere on your diagram. Okay, and then the last thing that I'm going to label are the SSBPs, or the single-stranded binding proteins. Let me underline this really quick. And then Okazaki. Sorry, last thing you're going to label is the single-stranded binding proteins. The role of this is basically to bind to ssDNA, which temporarily stabilizes it. The way that I remember what SSBPs do is that in my head, instead of saying single-stranded, I say stays separate. So that would be here. Again, I just say it needs to basically be situated somewhere up here. It would basically be holding these two strands. Helps them SSBP. It helps to prevent the two strands from basically like coming back together, like the two parent strands from coming back together. So SS or stay separate. Okay, so again, just remember that origin of replication is where that other half of the bubble would be. So imagine this exact image, but mirrored on the other side. Again, remember that 3 leads kind of rhymes, but remember that the 3 prime in the same direction as the direction of the replication fork movement. So that's why it is the green 3 and not this 3 down here, because the direction is moving towards the left, not the right. And then lastly, I remember that topoisomerase is at the top, helps to prevent twisting, so TNT, that might help as well. Twisting at the top. And it's also on the opposite side of the origin of replication. But yeah, hopefully this helps. If you have any questions, you can ask in class, ask in a session, ask on a discussion board. But I highly encourage you to practice. That is the easiest way to understand this. Definitely look over the definitions of like the roles of the different enzymes. That might help as well. And then here's a cleaner version of the answer key here. It's basically the same thing that I already drew, but here I have kind of like some of the things that I wrote up top on like how to remember things, little tips and tricks. So again, practice, practice, practice. That is the best tip I have for this. It's definitely going to come up in some way, shape, or form on your exam, so I definitely recommend knowing it, understanding it, and also flip it. So instead of 5 prime being on top and 3 prime being on bottom, flip it, because that will flip your leading and your lagging strand. It won't flip anything else, but it will flip that leading and lagging strand. And then I also encourage you to flip the actual image. So mirror the image, and make the opening, instead of going opening to the right, open it to the left. So have it basically go skinny on this side and then open this side, and then have your numbers whichever way you want them, 3 or 5 on top and vice versa on the bottom, just so you can kind of orient yourself and see that it is the whole picture. So if you do try that, here is the key. If you try this specific one, this specific example, again, it is the exact same thing, just the leading and the lagging strands are flipped. But if you mirror it, then everything will basically be the same, but mirrored. Again, if you have any questions, let me know. We will definitely be practicing this in sessions, and I highly, highly encourage you to practice this on your own when you have free time, just to test yourself. I hope this video helped, and I will see you in class.